Thank you, Sitabra, and that's it's a very wonderful opportunity for me to give a talk here. IMSC, this is my first time here. I have visited IIT Delhi previously, and occasionally go to Vellore through Chennai, so where my collaborators are working. So, yeah, it's a good thing that you are doing this. It's a very timely meeting as well. I think there is very few meetings that are happening in terms of brain dynamics, computation. It's a very niche field, I think, developing in India, and I think some of these meetings, if you continued more in next years, would be very much more welcome to the community and students. So, what I would again, like the previous speakers, I'm going to introduce a lot of concepts. It's a little concept heavy talk, so please feel free to stop me and ask questions. In kind of the theme of or conceptual thing that I think Sridhar was trying to develop in terms of structure, functions, how structure is related to function. That's what one of the things that he was mostly talking about. So, we will actually dig deep into that a little bit more. So, in terms of we are going to look at how representations. So, by representations, the first thing that comes when you hear the word representation, is there anybody who wants to define what representation is? It's like that, the conceptually it's something like that attention. We tend to have an understanding what representation is, but it's a very loaded term in neuroscience. To me, representation typically means a map, as simple as that. So, map is something like you think about today morning I was trying to walk to the beach, and I turn on the Google and I try to find a map, and then when I am seeing it, I am trying to make a mental map or mental representation of that Google map in my brain. So, that is a representation, right. So, representation, as simplistically speaking, is how we develop this kind of map, and this is how we look at data. I mean, the next stage you can look at that in terms of firing rates. Somebody talked about electrodes. I mean, you can look at EG, you can look at population, but at the bottom of it, some things that are going on, that is like this complex brain structure, which is this very complex electric circuit, is giving rise with some of these features to this representation, and that is what I am going to talk about. And for people who are from physics background and maths background, so one more thing that I am going to talk about, how representation can actually appear at different time scales, and this is something a very dear topic to me for a long time.
So, Akbar, it's interesting you mentioned that example of map. So, one of my first experiences when coming to Chennai, and this is before the Google Map era, is I was trying to explain to an auto driver how to go from one point to another point because he couldn't understand where I wanted to go. They folded out my map and I said, I want to go from here to I want to go there. And the thing is, I understood that he didn't know how to read a map. I mean, in the sense that he can see in paper, but he can't represent it in 3D. So I later read that apparently this ability to read maps is an acquired skill.
Absolutely.
You can't just give it to some person, there's no experience.
Absolutely. So, it's how to sort of consolidate these representations, and not only in terms of maps, and this is something that I am not going to talk about. Lately our lab has been working on how do we develop sort of in general imagery. So, for example, if you read a novel, for example, some novelists seem to give you a feeling they are very graphic, right. So, when you read that novel, you can actually live the experience. So, what is going on around you, like you feel that you are immersed, like a immersive experience, versus some novels it seems very fact driven. So, that is again a map, but I can go on and go on this, let us talk about it later. So, this is a piece of art, and it is one of the brilliant pieces of art by one of the brilliant mind of our times, I think Salvador Dali. If you ever visit St. Petersburg in Florida, it is a very strange place. You do not expect a Dali museum there, but it is the second largest Dali museum in the world, and this is a picture that is there. So, and it talks about these two sides of the brain. Remember, Dali at this whole age 1940s, 60s was living in a time when extensive development of brain mapping was going on, like this whole idea of this Brodmann, Penfield, I mean, all these maps were getting developed at that time. So, here he talks about, I mean talks in a very, draws about, sees things, right. as these two sides, two hemispheres of the brain, and at those times the right hemispheric brain is even till date is kind of a lot of people who are in neurologist discipline, they think that right hemisphere is involved in a lot of cognitive functioning, particularly analytical reasoning, this kind of stuff. Left hemisphere is more artsy type sort of thing, right. So, this maybe I always get this wrong, something to do with my mapping, but the point is, so there is an asymmetry, and this is something that I think Sridhar was also referring to in some degree in a very sort of in terms of the left versus right asymmetries that exist. So, this in the point being that this asymmetry what Sridhar was referring to is something that even Dali talked about in some sense, it is not so new again in a completely different realm, in a completely different kind of context. Now, of course, this is what we know, that there is this language areas of the brain, and these language areas were discovered by this guy Pierre Paul Broca in France, and this is the brain of a guy who could, like, due to inflammation, this particular area of the brain we call this inferior frontal area. I think there is a pointer there. So, this over the course of his life, this area, there was a lesion in this area due to inflammation, and what happened was slowly he lost the ability to speak most of the words, except he could speak one word, which is tan. So, he would just literally say tan, 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 and it was in the left hemisphere. Now, what also was interesting, this guy had no problem whatsoever with kind of language comprehension, meaning his auditory system, his ability to understand language is intact. What really was impaired is this ability to speak certain words, most of the words, right. So, in some sense there is an asymmetry again, that if it happens in the right hemisphere, you do not get this kind of a situation, but in the if it happens in the left hemisphere, you have this example of this guy tan man, and this again this brain is like displayed like a piece of art in and this is what the French can typically do in it is a museum in Paris. So, again if you go to Paris, do watch that brain, and it has a giant hole in the the inferior frontal areas. Now, this is another way of another study recently, in somewhat quite recent actually, a study which compares, it is a meta analytic study which compares several thousands of papers from different disciplines from, if you see it's sorry for the resolution and the small. So, basically what this picture depicts, I did not realize it would be so small and so bad resolution. So, about if you look at all the animal kingdom, so here is kangaroo, so grey kangaroo, all the way lemurs, macaques, there is humans in somewhere in between, yeah. There is pigs, domestic pigs. So, all these animals have different degrees of left-handedness, right-handedness and ambilateral. Interestingly, if you take the average across all animals, it is actually like split out in 1/3, like 1/3 of the animals are left-handed, 1/3 of the animals are right-handed, and 1/3 of the animals in the, or 1/3 of the entire population of, if you combine all the populations of animals and humans, they are ambilateral. So, in some sense again this laterality is somewhat innate in organisms, this asymmetry, this laterality comes from certain asymmetries, and we will definitely dig down where this asymmetry comes from, but this exists in the animal kingdom as well, as we go for different sort of different kind of species. Now, what I am going to talk about today, so how do we understand this laterality in the first place at a very sort of fast time scale, the time scale of EEG, time scales of brain waves, so to speak, and also from a much more slower scale, such as at the level of, which is also at the electrophysiological level, but it is a recently been found out at a very ultra slow regime, like it is like less than 1 hertz, and some of these rhythms are actually related to your internal attention. This is slightly different, I hope Sridhar would agree, but the attention that Sridhar talked about, as he said in the last slide, that attention had many different subparts. So, he talked about the sensitivity and bias. What I will talk about attention innate attention at a very slow time scale, which decides our fundamental sort of levels of performance, your baseline levels of performance, can also emerge from neural structure. So, this is the two things that I am going to talk about today. So, why is this interesting? So, if you look at the auditory system, and my sort of object of interest here is not the visual system like Sridhar worked on, but it is the auditory system. Now, as I said in the previous slide, that you have this beautiful example of like this Broca's area, the language areas, where you can find this left hemispheric lateralization. You also have, for example, if you are talking about, if you have a right hemisphere lesion on the other hand, what that will impair is your ability to understand certain components of music, melody, and this is something been recently shown. So, you can think of in the auditory domain, while the left hemisphere is allocated to some sort of speech sort of sound processing, language processing, the right hemisphere is processing something different features of sound. Some things like like how the frequency, FM modulated tones, because this is what music is comprised of, like FM modulated tones. So, this is what is going on in the right hemisphere, right. So, how do that sort of thing really appears, but on the sort of substrate, like if you look at the density of fibers, if you look at the how the two cochlear neurons are connected to the different sides of the brain, the left and the right brain, they look quite symmetrical. In fact, if you take the DTI of primary auditory cortex and how primary auditory cortex is connected to different areas of the brain, different like other areas of the brain, there is also enormous amount of symmetry. The distribution of fibers, the length of the fibers, the density of almost identical like Gaussian distributions. Yet what matters is somehow there is a change in this symmetry when it comes to function. So, function kind of sort of there is some sort of an asymmetry in the function, while there is some kind of symmetry in the structure. So, this is something that we are trying to understand, right. Now, and this if you see, this is another picture from a review paper from Carolis et al. So, this is again if you see that if you look at different axes, for example, of different axes of sound sort of processing, if you look at symbolic communication, emotion, perception, decision, for example, Sridhar talked about decision. So, if you look at this there is some sort of some things which are kind of, it can be kind of both ways, but as you go to this left dominant activations here, you have this very nice symmetry where it talks about this different kind of auditory action, happy, emotion, all this stuff, and then there you also have these expression, these are all right dominant sort of function. So, again, so there is kind of a map, there is kind of some sort of a gradient along these two axes of functions in the auditory representations. So, the fundamental question is, so if we talk about this is like exactly the same thing as the GPS example I was giving earlier. So, how do these kind of maps get set place in the in the brain? I mean, how do we develop this kind of intricate sort of dissociation of function? Now, a very simple, if you look at any psychology paper, I mean the way they describe it, well, it is kind of this kind of divided, you allocate like it is like a sort of an organizational behavior kind of example people or explanation people give. So, if I do some kind of perform a task in an office, I distribute the task to some certain subcomponents, and this is coming out of something like that, some kind of a division of labor approach. So, if I distribute all these different components to different areas of the brain, I will perform better, but why left and why right, nobody knows, right. So, essentially the thing that we are going to use for the purpose of this study, this first example, is that we are going to use EEG, where we are going to look at neural oscillations and try to understand from neural oscillations and relating it to the brain structure, how do these kind of representations are happening due to the physiological constraints. So, there are some work done in the literature. This is one paper by Peter Lakato's group in Current Biology in 2019, where they look at this cortical activity between the speaker and a listener, So, if you take the amplitude of the you take the speech sound from the speaker, yeah, speech is a FM modulated sound. So, if you look at, if you do a Fourier transform on your speech sound, you will go like, you know, the energies of the frequencies will be distributed in some form like within an envelope like this. So, it essentially means that there is a very high pitched sort of change here, then it kind of quietens out, then again then you have some kind of slow modulation over the entire range, and again there is some kind of again pitch change, and this way and so on and so forth. Now, if you take the envelope of this Fourier spectrum, right, of the speech, and if you look at the EEG of this like, you know, activity in this listener, for example, the envelope of this speaker signal is somewhat correlated with the brain wave of the listener. So, in some sense there is some kind of a slow rhythm that is kind of we are trying to, with which we are trying to integrate the speech signal into to understand the meaning out of it. So, one of the knowledge, and I think this is very represented very like, you know, there are, maybe I can go back one slide back. So, one of these things that we think about, I can, so there are these two areas of the brain, right. So, this is our auditory area, which is also called the Wernicke's area, and this area is called the Broca's area, is the speech area. Now, how the sort of, you know, sound converted from the sound representation to the meaning of this words and letters happens because of the interaction between these two areas. So, this is something that also you have to keep in mind. So, when you talk about this kind of, when I show this kind of results. So, now this is for complicated stuff as speech. Now, what happens if you want to understand speech then? So, you really need to understand how does even a small frequency or just a basic fundamental frequency is represented in the brain. Now, there is a kind of, you know, this is very popular way to look at the brain itself overall. There is ways to, you know, entrain the brain into certain rhythms. So, for example, if you present at certain rhythm like for a long time, like you have to present it for at least 5-6 seconds, like, you know, signals at a certain frequency, maybe 20 hertz, 40 hertz and all that, what you can generate a frequency in your brain at that frequency, right. So, at the same frequency which you, it's kind of, you can think of it as a resonance is happening. In physics you have this concept called resonance, this is something similar, right. So, and you see a demonstration of that. So, if we present in this case a 40 hertz sort of sound, so meaning that I have a tone which is kind of, if I decompose it, it looks like this, it is a 1000 hertz tone, like basically it sounds like beep 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 like that, right, but I am repeating it 40 times a second, right, and this is the building block of any kind of sound, maybe music and speech. So, if I want to study the representation of something like that, this is what it looks when you see the EEG activity. So, this is when I put the electrodes, this is what the EEG activity would look like. There is an onset of stimuli, there is an initial burst of activity which we call the evoked potential, and what you see, this is what is the entrained phase where you see the 40 hertz sort of rhythm. So, the same rhythm which you gave as the stimulus in the auditory stimuli, you are generating the frequency at that same rhythm like this, right, in the EEG. So, in a way that you are tuning, right, you are tuning your brain, like, you know, like you have the principle of radio. So, if some signal station is giving some kind of emanating some sort of frequencies, and you tweak your radio sort of, you know, tuner at that frequency to receive that signal at that frequency, which you then gets amplified. So, if you think about it, this some similar kind of operation is happening here, right. So, there is some kind of tuning your brain does. So, something like what you do with a radio, in hearing a radio station, it is hearing out the signal outside and it is kind of tuning the brain signal, so to get it entrained, so that the rhythm is getting entrained in the brain. Now, if this is a mechanism that the brain does, how does it really do it? So, that is the question that we were really interested in. Okay. So, if I can tune the brain at like some kind of frequency, what the brain really has is this physiology it had. Somehow it has to use the physiological complexity to get tuned in that frequency that is being presented from outside. So, this work actually is done by my PhD student Neeraj Kumar, and is part of his PhD work. So, and if you see again, this is the sort of what we did at these different representations. We did it in a bunch of ways, like a scientist would do. We presented this tone in 1 year, 2 years, and then put it bilaterally with both ears, and we looked at how this frequency power, like the energies in this frequency which you generated in the brain, change across these contexts. And lo and behold, in the binaural case, you can take the monaural left and the right, meaning that you are presenting to each ear, and then you sum up those monaural left and right, you can get the binaural. So, it is like some kind of energy summation is happening in the brain, right. So, when you and this is really looking at the interphase clustering, but this is also the raw spectra. So, there are two aspects to it. Now, when the brain is getting trained, right, at this frequency that is kind of being generated outside, now it is exciting. So, there is the neural population, if you think in terms of the neural population and the firing rates and what not. So, there is an entire neural population which is kind of engaged in that activity, but an Indian population also needs to involve actual different information from different areas, and they need to come. If you now this within the neural population, you can think about different kind of Hodgkin-Huxley neurons are there, right. So, some of them, all of them together have to get in phase together. Some kind of synchronization phenomena has to happen, and that synchronization phenomena which is happening has to increase at the presence or during this entrainment phase. Now, the way that we can study that, we can look at the phases of this signal. So, if you look at different electrodes in the brain and you can somehow map all these sources, and there are techniques that I will just briefly touch about how you can do that. So, you can sort out the phases of the signal at different locations of the brain and look at a measure called the phase locking, which basically looks at the phases of the signals at different parts which are related or connected to the auditory areas. So, what I am showing here, that not only that individually at the electrode there is an increase in power at 40 hertz, but there is the brain has this tendency to get in sync, right, across different areas. So, the signals across these different areas are phase locked at that particular frequency. So, not only there is oscillation that is being generated at specific brain areas, but the brain areas are getting integrated in some sort of a cohesion when this kind of signal is getting entrained. Okay. All of you get this, okay, conceptually?
Is there a speed sound being played over the 40 hertz?
No, this is a simply tone, no speech yet. So, the reason that we want to do this, it will get clearer. Why do we want to do this? Because this 40 hertz is very important, because you need to understand, this is, as I said, it is exactly the radio. In the radio, so you have, if you have one frequency, you can easily tune to that frequency. But think about the speech now, as Sridhar pointed out. In speech, I have a very complex sort of a distribution of FM tone. It is very difficult to tune it to one frequency, because if I to look at one brain frequency. So, speech will have a representation, but it will be multi frequency, and this is what I will get at. So, yeah.
If the input signal is, let us say, order of magnitude higher, maybe 10 kilohertz, you still expect this
Input signal, you mean, yeah. So, you cannot go certain beyond, because if you do the, well, you can increase. So, your sound level is between, like our 20 hertz to 20 kilohertz, you have to be within that frequency, but you cannot be, there are I think 2 points to that question. One is you increase the frequency, yes, you, as long as frequency increase does not matter. You present the frequencies at that same 40 hertz, you will get the effect. You can change the 1000 hertz tone to 2000, 5000, 10,000 kilohertz tone, it will remain the same. You will still generate 40 hertz, because I am repeating it at 40 hertz. The second thing that you should keep in mind is that the decibel, which is the amplitude of the overall signal, right. So, that we cannot exceed too much, because if we there is after 90 decibels, I mean our ethics will not allow that, and your cochlea would not like it. So, it will have.
What if I use a low decibel? It's not very high.
It is a medium decibel, around 60 to 70 decibel we use. We do not go above 90, that is very loud.
Yeah, if I use that at 50 decibel, but I jack up the frequency to 10 kilohertz.
No problem, you will still get 40 hertz, you will still see 40 hertz. The 40 hertz is coming, there are two frequencies, okay. So, if you see there is a jiggle here like a high frequency, this is what you are referring to, I think the 10 kilohertz part, this part can be 10 kilohertz, no problem. This repetition of the 10 kilohertz, this is the one is the carrier frequency, one is this 10 kilohertz frequency. So, 10 kilohertz frequency which is sampled at 40 hertz can still generate an effect at 40 hertz. The brain rhythm does not go to 10 kilohertz, because the problem with that is maybe it does, I do not know, but whatever in terms of neuroscience of EEG and even to some degree in spikes, everything above like this 100 hertz or 250 hertz is basically we think of it as noise. It is very difficult to distinguish it from noise. So, probably it can go there, but it is difficult to measure, I do not know. But typically what we do to understand the entrainment is that we repeat it at low frequencies. The low frequencies can be easily entrained and we can easily see, because what we believe typically your noise floor in the EEG, anything above, at least in the scalp EEG, anything above 60 hertz 70 hertz is noise. If you are recording directly from monkeys or humans, as we do in this something called this stereo EEG, which is when these epileptic patients have implants in the hippocampus or like different parts of the brain, there you can go up to 250 hertz, but that is about it, okay. But that is this is the range, we can't go to 10 kilohertz and measure signals in the brain, that will be considered as noise. Maybe there is signal, but it kind of gets mixed with the brain noise, okay. Any other questions? Yes.
Is it like wave packets that is?
Yes, so this is exactly a wave packet, correct.
So that within the wave packet the frequency is like 10 kHz?
It can be 10k, it can be 20k, it should be the hearing threshold. Hearing threshold we can hear up to 20 kilohertz. So, you do not want to go very high, because it is very, it is high pitched kind of sound, so it is kind of uncomfortable. So, if you go too high.
But the wave itself propagates at 40 hertz frequency.
Yes, correct. So, it is modulated at 40 hertz, correct.
So just
Yeah.
if the signal is not modulated, as if you take a pure tone, just one single frequency.
Just continuous 1000 hertz?
Yeah, not 1000 hertz, let's say 40 hertz, but pure tone, no modulation of the signal.
No modulation of the signal.
So for example, this is modulated.
Right. So, but how do you change it? So, you still have to have some sort of if you want to make it 40 hertz, you have, so see there is this sound, I am tapering off by some kind of smoothing out these phases. So, you still have to taper it, right. So, how would you create that 40?
Just a signal with just one frequency, no modulation of the amp. Just one.
No, this
sine
is one frequency. The frequency within this, the frequency is two frequencies. It is not modulated, but you have to have a frequency. Otherwise, how do you hear? So, it's a wave, right? So, which you are hearing.
So can I generate a signal which is pure sine wave?
This is pure sine wave. This is a sine wave. Within this, there is a sine wave. Now, if what you are asking, let me draw it in the board. What you are asking. So, you do, but there is a problem with that. That is what I wanted to say. So, let us say you have this sine wave, right. You have something like this, right. You get 40 hertz. There is the one problem with that. In the perceptual space, we get something called a click artifact, meaning you will like have the tone like the perceptual you will have an additional click click click, something like this you will feel. It does not exist, like illusion. So, to cancel this out, we actually make this like this. This is still a sine wave, I am just tapering out some kind of a linear function to it, so that it kind of tapers out. It is still a sine wave, there is no FM modulation here, there is only two frequencies, one is inside the sine wave and there is another sine wave outside, clear? Because if you keep it just straight like that, there will be this click artifact, which is kind of, it is like a CR. So, these perceptual experiments are a bit difficult, so you have to take care of lot of things to make sure that things are coming out as it should, because this perception has, as Sridhar pointed out, there are a lot of these biases. So, these click artifacts, this kind of illusion similar to that visual sort of space, like the visual, I think what Sridhar was trying to point out, that we make out like there is some kind of a reconstruction going on in the visual space to give you a sense that it is always fully loaded, but you are not really attending. Same thing happens in the auditory sort of domain also. So, you think that is you can hear out everything, but you are not really hearing out. So, some things that brain is filling out and making these illusions. So, if you keep it like this, so this click click click, these are actually illusions. So, this does not exist. Okay. Okay. So, now coming to this is the spectral power. So, what we can do is basically when we have EEG like this, we can take the MRI data and we can co-register the MRI, and this is something that I am not going to too much depth, because it is itself a complicated way of doing things, because what we typically do is a lot of these algorithms came from radio astronomy. It is this technology called beam forming which we use. So, the idea is that if you have let us say imagine that you are getting light sources from these different stars, and you have telescopes at finite locations in the planet, right, but the length and the distances where the light is coming from you do not know, right. So, in some sense in the brain we have a similar problem. We have infinite number of locations where the signal can be coming from, and we have some few of these locations where we have electrodes. So, we have a inverse problem, that where we want to be at the source we really do not know. So, what we typically do, we do a kind of this whole like class 12 electrostatics problem, like the Coulomb's law. You have a charge carrying conductor inside, you can compute the what is the Coulomb's forces outside the head. We use that to make a forward model of this electrostatic potential, and then we reverse it and we kind of do the least square feet, and after that we get this kind of images of the brain where we look at the source activity. So, it is computer generated, there is a lot of softwares nowadays to do it. So, it is little straightforward because of that, but you can do this. So, now after you do this kind of images, and these are not MRI images. So, this is MRI is being used to guide the source localization better. So, you have individual subjects MRI, and what we see in the case of the monoaural left and right, as you can see, we could generate activity in the entire auditory network, the auditory cortex and all these other areas. And interestingly, and this is for the purpose of this talk going back to representations, what should be interesting to you is this binaural case where we have sound in the both ears, and even visually what you see, there is a right hemisphere dominance of brain activity. So, meaning functionally, so even the structure of the auditory cortex are, I mean size wise in terms of network like structural connectivity wise they are same, both the hemisphere. Functionally there seems to be a whole large amount of activity or the sources in the brain that are active when you have a completely symmetric sounds being played to both ears. So, there is some kind of innate kind of lateralization that we want to talk about, that is present. Now, if you remember, the Broca's area was on the left side, that was it was first discovered. Here we are getting for the tonal sign sort of an input, we have a right hemispheric dominance. So, it is a little bit opposite to the one that you see for speech kind of sounds, and this is something that kind of will come back and forth, and we are not the first one to discover it. So, the people have talked about it before, Robert Zatore's group. What is interesting though, this is something what I computed this earlier, no, a more sort of mathematical way to compute whether it is lateralized or not is to compute something quantity called a laterality index. In simple terms, let us say that you have two hemispheres in the brain, you have two analogous area, like this is the motor cortex, this is the motor cortex. I take it the motor cortex in the left hemisphere and I take the motor cortex in the right hemisphere. I subtract the activity between the two and divide it by the sum of the activity. So, that is my lateralization index. So, if the left hand side is more activity, so it will be sort of if it is LH minus RH. So, this is going to be like the positive sort of a laterality index. If the right hand side is more, if you do LH minus RH, then it will be the opposite side is going to be the it is going to be, if it is negative, it is going to be the right hand side. So, typically we play with the RH minus LH to avoid this confusion. So, in the if it is negative laterality indexes for my figures that I will show, it will mean that the activity is left hemisphere dominant, and it is if it is right to the positive, it will be the right hemispheric dominant. So, if I plot this and if you see this, that in the binodal case it is slightly above 0. Now, in the monodal left and right, and this is for a something called, and I am computing this for something called the evoked potential amplitude, which is the latency that is the first volley of activity. So, remember that when I showed you that first time I showed you how the stimulus look the response look like, there was this big volley of activity like which is I called the evoked potential, and then there was this 40 hertz steady state activity that appeared. So, this is from that part which is the initial volley. So, this is just to go back here, this phase. So, this is called the N100 kind of amplitude. So, I am measuring whether left hemisphere or right hemisphere together how they are contributing each area wise. So, I am contributing to the laterality index. So, I am going to the auditory cortex and I am computing this laterality index. Where did I go? So, what I see here that it is very contralateral, meaning that if I take the left hemispheric sort of if I put the sort of sound in the left hemisphere, the right hemispheric laterality is more. If I put the sound in the left right ear, the left hemisphere is more, but that is only for the auditory cortex, the primary auditory cortex, and this is what this shows. And but in the case of the binaural, what I see that there is a more activity is biased towards the right side, and this is something which you guessed from the sources probably, that if it is towards the right side, if I take the average across all the brain areas, it is kind of should be reflecting. So, it is kind of reflecting this result here. So, the reason it is positive because there is a rightward bias of this activity. Yeah, everybody understand this? So, this is little bit above 0. So, this is significant. So, overall there is a rightward bias for something like a 40 hertz sound. Now, the question is this ASSR representation, if you tune in a particular frequency like the radio, you start to get this very rightward asymmetry in the brain. Now, is this effect more at the network level? This is something we also see, like if you look at more complicated measures, here I showed you only the measures like phase clustering and also this phase locking, sorry. Now, what we can also do, we can look at we can build these networks among these different networks of the auditory cortex. We can do something like a causality measure like Granger causality, and we can show that if you take in these higher order network measures, that it even gets this bias gets even heavier. So, meaning there is this network level there is even more bias. So, now what happens to speech and music, and this is the way that we want to question. Yeah.
So, I am going to throw the same question back at you that you asked me. So, does this have to do with handedness, the right laterality? The participants equally left and right handed?
Yes, we do not know that yet, but it is something that we do usually, like the speech and language, typically it is not dependent on your handedness typically. That is what people have shown. I mean, we did not show this in our data, but overall people do report that it is not dependent on your handedness, because there is it is something that overall effect. But now, is it true that all circumstances you will have speech on the left hemisphere? What happens, for example, if somebody has a stroke in the left hemisphere? So, what people have also shown, when if you have like people with having stroke in the frontal cortex, stroke in the inferior frontal areas, it seemingly the right hemisphere helps in recovery. So, there you start seeing that higher activity in the right Broca's, it kind of shifts around. So, in some sense there is an advantage. So, the right Broca's area exists, it is just not being used for language, and it is not completely 0, the activity is not 0, it is there, it is just that it is the left is dominating. Now, the reason that why it is so, and there is some reason that we are seemingly trying to uncover, and the way we will try to uncover this, and this is something that somewhat down the line when we were working, we started our lab working on this auditory sort of steady state. There was this paper by Zatores group in Science, which developed this set of stimuli, which is I think brilliant in some sense, to use it for this kind of questioning. So, Zatori, Robert Zatori is a very senior professor in McGill, and he has been looking at this lateralization in brains at different species for a long time, last 40-50 years. So, what they developed the stimuli, particularly this Philippe Alboy, his postdoctoral fellow, is that if you can merge, so if you can create certain stimuli, and this is, I do not know if I have the sound here, so it sounds like this.
The children's dog. The servant came to love the children's dog.
So, it says the servant came to love the children's dog. So, these are called this a cappella song. So, as you see this melody, so now I can use the same melody to utter this a different lyric. They said it was a cross they saw in church. So, for example, the melody will remain the same, the lyrics will be different. Now, I can also change the melody and keep the lyrics intact. In this case, that can be also created. So, these a cappella song essentially have this base set of melodies to which you can put these different kinds of lyrics, and you can crisscross them, and what you can develop, put them in terms of
The servant came to love the children's dog.
in a delayed match to sample task, and this is something like a force choice task, but what you have to do is that you have to kind of, it's also have a match to sample component, meaning you hear the speech, so you retain it in your working memory, and there is a song 1, there is some kind of in between, there is kind of an interstimulus interval. There is also a song 2, which is that they came to the servant came to love the children's dog, or they went to the church and what that. So, these two songs are being presented in sequence with a gap of 1 second. Now, there will be a question that in some cases if the cue is speech, then you will say they are same or different based on their lyrical component. Okay. If the cue is melody initially, so you will say whether the music is different or the melody is different or not. Now, the idea that you can see where they are going with this, the idea is if you have speech, the speech should give you a representation. If I have this speech component, if the task context is speech, I should get for the same stimuli left hemisphere activation, and if the component is sort of music melody, then for the same stimuli I will get the right hemispheric activation. So, this was the goal of that study. Okay. So, now you can see this, right, why they are designing the stimuli, and lo and behold what they saw, that interestingly, and this is something, yes, I will come to you. So, they took at this this area in the auditory cortex, and there is this way to analyze the data called this MVPA, multivariate pattern analysis. What it essentially means that instead of looking at like one voxel, you look at multiple voxels within a defined area when you get the MRI data, let us say 1000 voxels, and instead of tracking one voxel activity, you look at a pattern of activity, and it is basically like something that what Dipanjan also used, this some kind of you can apply simple like classifiers to do kind of this discrimination, whether you have a pattern for the left and the right. And what you find out, and they particularly looked at this anterior part of the primary auditory cortex, not even the Broca's area, seems to be more active. The primary auditory cortex is more active when you look at this detailed MVPA analysis for the sounds when they are like in the speech context, when the brain activity is in the like triggered toward the identifying speech, versus it is in the right hemispheric part of the auditory cortex more is active when there is the context is to discriminate between the melody. So, in terms of it does not even need to go to the language areas. So, the entire thing is happening, this discrimination is happening at the primary sensory level. Let me allow me some time to sink that in. This is big, because till date for the last 100 years neurologists and everybody was thinking about that this whole like decomposition is not happening at the primary sensory level, but this left versus right lateralization of speech versus melody or sound is happening at this higher order cortical level, at the level of the Broca's area. But what they are finding here is dramatically different. that even at the primary cortical auditory cortical level, there is this dichotomy that is visible between the lateralization. So, which is why it went in science, it is a huge result. Now, what it changes is that the way we see about this lateralization. So, in some sense, and this is something Ladas and then they went on to propose, the reason that is happening left versus right is the first time we came out with a mechanism that why this might be so. And one of the hypotheses they proposed is that what is really happening at the functional level is that the requirement of the task, in this case for the speech, which is to discriminate between very fast spectrotemporal changes. When we are hearing somebody speak, we are trying to hear out we are trying to piece out differences in fast energy spectral energies, which are very finely sparsed in time, versus when we are talking about melody, we are trying to integrate things that are like situated much longer. So, like a low drum, kind of a low beat you can think about, right. So, what they are saying, this requirement of this fast versus slow time scales, which are the requirement of the functional sort of requirement or the functional requirement, is dissociating these two pathways versus the left and the right hemisphere. Now, what we wanted to find out, what are the underlying neurophysiological marker or factors that can change this. Now, you had a question, sorry.
Okay. I was coming to like these are high order functions.
Yeah. Okay. So now Neeraj came out with this idea, like in biology there is this whole genes having different functions. It's called this concept is called pleiotropy. So is it possible this functional degeneracy that we are seeing is coming out from the same structure by some kind of a trigger mechanism, something like a switch. And what we proposed that there is some kind of I will just keep this, no I can't skip it. So, there is some kind of a delay triggered switch, because when you are sort of what is really happening, the underlying structure that this is the components of your brain are connected via some time delays and coupling. And this is how this some kind of a delay triggered switch is operational to kind of give rise to this lateralization, and I will show you how much time do I have. Okay, I do not have much time. So, okay, so we record the same experiment when people were doing EEG. Now, what we look at is that we associate we found out the primary auditory cortex, and we looked at the connectivity, and the way we look at the functional level is looking through this net Granger causality analysis. And what we found out that we took the auditory cortex as source, and we look at the entire brain kind of Granger causality from the auditory cortex, which was activated for the areas which was activated, and we found out again the same kind of results at the network level. What you see in the speech case, so speech context, and this is the melody context, and this is the 40 hertz. So, the 40 hertz kind of results reflect the result that was before at the individual source level, but here we are looking at the activity from the primary auditory cortex to the other areas, the Granger causality, it is slightly different, it is a network result. So, again what you see there is 40 hertz, it is more or less bilateral in the case of the Granger, but there is some kind of activity also spanning both sides. At both these kind of inferior areas there is some activation. But what really is very amazing that we could separate out that at the delta and the theta regimes, if you see this activity, the left hemisphere is more dominant in the speech context in the delta and theta, but in the melody context the right hemisphere is more dominant. So, again the results which were replicated for the primary auditory cortex with MRI, we could recover the entire auditory network at those speech and the melody time scales with EEG. So, but interestingly when we look at these higher frequencies like beta and gamma frequencies, which are typically at 15 hertz and more than 30 hertz to 40 hertz, we do not see this dichotomy. This exists only at this delta and the theta regime. Now, we take this further and we can as you can see we can look at all these different brain areas from superior temporal, pre-central and all these to parahippocampal areas, and we look at for the 40 hertz and the delta, theta and the beta and gamma. Again there is, as ASSR was expectedly right dominant. So, in each area we compute the laterality indices. So, and as you see the positive, as I said, if it is towards the right, it is right hemispheric dominant, if it is towards the left, it is left hemispheric dominant. And as you see that, you can see the delta activity here. For the melody component, again things are more right hemispheric dominant. For the speech component, it is all left hemispheric dominant. So, this again this result kind of another way to sort of demonstrate this result. Now, how do we really sort of develop an understanding intuition for the how the structure is shaping this up. So, we take the same kind of data that Sridhar explained. So, and thankfully I do not have to explain again. So, we can compute the with some kind of brain parcellation what is the length, which I think you did not really look at, but we can we compute two things, we compute the length, we also computed the density. So, the density can be translated to something like a structural connectivity matrix, the length and density together, and this can be weighted to get some of these, each individual area can be thought to have different kind of weights based on the density of their connections. And this is, and people familiar with this, I think a lot of you are familiar with the differential equations in this crowd. So, we use something called this Kuramoto oscillators, which is a phase oscillator in which we can model the phase of an oscillation via a very simple sort of term, which is this sin theta, and there is a coupling which we can use, which is this global coupling. And there is this individual coupling that comes actually from now actual DTI connectivity. I can set these oscillators up at all different regions where my structural connectivity takes me, and I connect them via coupling, which is again derived from diffusion imaging data. I can also use the time delay factor, which is scaled to the length of the fiber that measure divided by some kind of velocity measure that is again physiologically derived. So, we can make a detailed biophysical model of how these phases of these oscillators are going to be connected. Now, what I am going to do, what I fit, then the next stage what I do this is that at the next stage, and this is something we have done in a lot of fanciful ways in another paper with Victor Jersa also. So, but in this particular case we are using a simple maximum likelihood based fitting to fit this kind of model parameters, the frequency, the tau and the k. Now, that typically what we can have, we can set the tau is based on kind of 1 by conduction velocity, and this is something varying from 5 to 20 millisecond, and these conduction velocities can specifically change at different trials when the speech and the this context comes into play, speech or the melody. Now, I can fit this, and whatever I get, ultimately I take the, I use the laterality index as a constraining factor to fit all these other parameters. Okay. So, the laterality index, what I want to do, I basically look at the EEG data, I compute the laterality index, right. So, that is my empirical laterality index. I take the structural data, I put the Kuramoto oscillators in the structural data, and I predict the functional data using the structural data, and the way I fit them is via maximizing my best fit laterality index. Where can I best fit the laterality index? If I fit these entire model parameters in this way, what I sort of get is something like this. So, these are my sort of predicted sort of values of tau and k, and this is the correlation between the real data. This red line represent the chance level description. Interestingly, at delta and theta levels I could not predict very well, but at the beta and the gamma I could predict the laterality index very well. And if you can see at that level that these things are more believable, the tau that I discovered from this high correlation are most likely to be correct. And again this frequency I can at this beta and gamma are matching, in the actual beta is around the 20, actual gamma is around the 40, which give my credence that whatever we are doing, the fitting is actually working. And these taus that we are deriving from this model are something interesting that we are saying in the next phase, that this again these are again correct prediction. I will skip this in the interest of time. What I could get to cut the long story short, we can get for this melody context, the time delays that we generate are slightly, as you can see, are higher. For the speech context, the time delay for these oscillators to be in sync together are much more shorter. In some sense, there is a kind of a delay triggered switch that as if the neural system is interestingly managing the conduction velocity based on whether it kind of needs to discriminate between very fast spectrotemporal differences versus something which is trying to integrate over a long time. And the way to do it will be to change the delays between these oscillations or the conduction velocities or the conduction speeds of neural propagation. So, this is something again what is interesting, there is a hierarchy. So, if I am going from superior temporal to like frontal areas. So, for example, sensory to higher order areas, my ability to kind of predict actually also deteriorates. I can get this kind of prediction of laterality index much better in the sensory areas versus in the higher order frontal areas it kind of deteriorates. This is something because again I think the higher order areas are much more bilateral in nature, essentially that is why it is happening. So, again this is this what we found out is that dichotomy in the auditory processing can be related to time scales of information processing required, and essentially we propose this tuning by delay kind of a hypothesis that the network needs to can use like a radio to operate in a particular mode to kind of do certain kind of functional aspects of the task. And the delay parameters can be set by the structural context. For example, constraints is the myelination, it can be also functional constraints like cholinergic inputs, conduction speeds and synaptic scaling, which are developed over time over learning. For example, one thing can be possibly to look at is how musicians kind of react to this kind of stimuli and how their kind of conduction speeds and synaptic scaling has changed and what kind of values we can extract for conduction delays when we fit their laterality indexes. So, these are all interesting questions. You can actually start out with this kind of paradigm with how once you can start model these things to these kind of models, these large scale connectome models. There is one more thing, if I have 5 minutes I can talk about, since I promised Sridhar that I will talk a little bit about attention. So, so what we are interested in, so trying to show you that similar kind of modeling different time scale and can also give an understanding of some of these recent discoveries in the attention. As Sridhar talked about, there are different ways to look at attention and different levels it can emerge. So, in this particular scale I will be talking about attention which is kind of very endogenous in the sense that it is kind of happening in your brain all the time when you are alive, from the day you are born to the day we die, this whole rhythm is going on, and this is something which Peter Lakatos in this particular work in I think 2016 in Nature Neuroscience first discovered in monkeys. So, what actually I should have another figure and to show what the supragranular and laminar alpha means. So, basically if you look at, so using a laminar electrode, meaning these electrodes are very special electrodes which can record from different layers of the brain. So, as you can look at go to the different layers of the brain. So, you can start getting very interesting recordings by recording at individual layers. You can measure the current densities by looking at these partial derivatives of this activity from different layers. So, a lot of interesting things you can do. So, what they did was that, to cut the long story short again, so they found the presence of a very slow rhythm, and this slow rhythm is kind of correlated with the alpha. So, alpha meaning that if you have again this idea of this carrier frequencies here, I will just plot it, I think it is easier that way. So, if you imagine that there is some kind of rhythm in the brain which is going at a very super slow speed, and there is this alpha rhythm which is at 10 hertz, and this is going at around 0.07 hertz, and this alpha is bursting here when you are at the peak of these rhythms. And if you present something, a stimuli at those phases of the alpha burst, you are not going to get very good performance of the task. But if for example Sridhar places his bias and attention, bias and selectivity, sensitivity sort of task in between these phases, your task performance, your reaction times, your ability to make those hits are going to get increase. So, if you are here, if you present the task here, to make a hit is going to be much more difficult. So, your sort of false alarms will increase. So, in a nutshell, so this is kind of this is something, an internal rhythm of the brain. So, this is what they discovered. So, recently in humans, people have discovered this similar kind of wave also exist, and again there is some kind of again it is peak to this alpha. So, this when there is an alpha burst, so if both humans and seemingly monkeys have the same way to have an endogenous attention wave which is correlating with your performance. So, one of the questions that we ask based on this so we call this low coherence mode in some sense. Basically what is happening at this kind of this coherence is increasing at this alpha burst. So, this particular slow wave is highly coherent with your alpha. So, we call these the slow coherent modes, which is giving rise to a very interesting feature of the brain. So, one way to think about attention, I mean it has very much similarity which Sridhar talked about attention. For example, when we talk about attention, what he calls the decision. So, a lot of people think that this is something is in a different way, we call this the appraisal of different evidences. So, there is the attention spans, like you gather evidence and then you make an appraisal. You gather evidence, you make an appraisal. Look, all the time you are doing this because you are predicting the next environment. What is going to happen? What is going to happen right now after you end this lecture? What is going to be the, will there be a tea or coffee outside? What are the kind of cookies? So, all the time we are predicting and then we are gathering evidence. So, this whole process goes along in some kind of innate sort of way. Right. So, this is what this is doing, and a lot of people think about at the phases when you are actually making those appraisals. So, there will be an increase in the integrative properties of the brain. So, there is more amount of information is being integrated versus when there will be like these phases when you are actually sort of gathering evidences, there will be more segregation in the brain. So, there is this whole notion in the neuroscience called functional segregation and integration, how we put different kind of maps is something considered as segregation versus how we gather all this map information together to make a decision, make some kind of compute some decision variable will be integration. So, this is something which is going on. So, in some sense this low coherence modes are involved in this whole process, this perpetual process. So, what we did is that use again a whole brain Kuramoto kind of model taking DTI data, and this we did for both human connectome and monkey connectome, and we could replicate that this only the human connectome and the monkey connectome because of some special properties could generate these slow waves, versus if you just take a randomly different kind of same number of nodes, if I connect them randomly or some kind of Lorentzian fashion, I am not able to generate these nodes. So, essentially I will just skip this a little bit, so we could actually derive these very beautiful analytical expressions why this is the case, because if we choose these natural frequencies from Lorentzian distribution, there is this in the nonlinear theory, there is this idea of this Antonsen Ott ansatz. With that we can actually show that the order parameter, which is the variable which is looking at the global phase of these oscillators, that is non-oscillatory anymore, it kind of becomes a real valued equation, and hence there is since it does not have any complex eigenvalues, there is no oscillations of these order parameters. So, we do not get this low coherence mode to be oscillatory in any other kind of idealized network, if like all to all networks or some kind of random networks. The reason that the brain is operating, brain is doing it, is because at least you need two modules. Now, these two modules at the very simplest of terms can be those two hemispheres of the brain. So, which needs to be differentiated in two different frequencies, and this is, and again if these two hemispheres of the brain is actually having the same frequencies, you do not get these slow modes. These two hemispheres of the brain has to be differentiated in certain frequencies, it is to be separated by a certain frequencies to get this, because this whole thing comes as a difference between the intrinsic frequencies of these oscillators, the combined frequency of this low coherence mode. So, which we can actually show. So, essentially what we are trying to say, that the reason the brain is modular and the brain is also hierarchical gives rise to these properties that you can see these kind of slow coherence modes, and I will be happy to talk about it later. I am just skipping a lot of it in the interest of time, and you can see that how in both sort of so this is actually experimental data from macaque and humans, which we are modeling with this white line here, and we can very beautifully show for both these how these things are generated. So, because both human and macaque connectomes have these properties, which is it is highly modular, it is hierarchical. So, because of these differences which is being created because of these intrinsic frequencies, which is this intrinsic rhythm which is coming, because if you think about very minute differences between the microstructural sort of connectivity in this which is happening again from the structure. So, it is the structure is related in a very roundabout way, which is giving rise to this kind of different frequencies, which is finally giving rise to these two different modes. So, that is it. So, for to conclude that this kind of modular hierarchical structure of the brain is very important for generating this kind of slow coherence mode, which is coming at a very slow time scale at 0.07 hertz. Again, why this is important? So, it is somehow important in terms of the very fast time scales, this kind of modular architecture, the left and the right symmetry, where we can actually tune this via delay to different parts of the networks, again to facilitate the division of labor. So, what I told you is a story is essentially how different kind of models you can use, large scale connectome models, to understand the different time scales, how representations are structured. I hope you find it useful. Thank you.